This is the complete absence. This is the normal brain scan. And then there are lots of individuals who have partial ACC. Some people call it hypogenetic or dysgenesis. And you can see here that it's not only small, but it also only seems to occupy sort of the very front aspect. Whereas here you see that there's a corpus callosum that I'm outlining that is goes across the same trajectory as the normal one, but is substantially thinner. And so again, we would just call that thin. Early on in development, the corpus callosum is thin, so it does grow over time. It grows thicker, but the number of nerve cells that pass through that location doesn't really change. Okay, and then for some individuals who have absence of the corpus callosum, the question one could think of is, well, where do those fibers, those 200 million axons, where do they go? And it looks like here is a potential explanation. These are called Probst bundles. And so normally, if you look in this schematic here, the corpus callosum crosses the midline like so, and underneath the corpus callosum is this structure here and the equivalent one over here. Those are both the lateral ventricles. Even though they're lateral, they're very actually close to the middle of the brain, and they are underneath the corpus callosum. But you can see here in this individual, the white sort of comma-shaped structures are the ventricles, and so they've now been pushed to the side. And instead of the corpus callosum crossing the midline, you have two parallel structures, these Probst bundles, which are essentially the fibers of the corpus callosum that are moving. If you could see it, they're essentially moving in and out of the plane of the picture. A lot of individuals with ACC have enlarged ventricles, particularly in the back of the brain, and the name for that is colpocephaly. And again, so this is the same kind of scan that I showed you previously. You can see the child's nose and his or her lips, and that picture's in the midline. And you can see this enlargement of the ventricle space here. And then you can look at the brain at a different angle. This is just what we call an axial image. This is the front of the brain here, the back of the brain, the right side and the left side. And this teardrop-shaped structure is the lateral ventricle. And they always dilate, when they dilate, in the back. And the fact that this part in the very front of the brain is not dilated tells you that it's not under pressure. So it's not what we would call hydrocephalus, which is where there's too much fluid and that fluid's under pressure. It's rather just enlarged ventricles without pressure. And it's important to know that because the child who has this condition doesn't need a shunt, which would be different if they had hydrocephalus.
Sorry, can I just ask a question there? Because our son, was born with hydrocephalus and complete ACC, and I was told that they were connected, like the absent corpus callosum and the hydrocephalus were not connected. This is the first time I've heard anything about this.
Right. So hydrocephalus and the absence of the corpus callosum are not connected, but these large ventricles, I'd say, show up in about 2/3 of individuals who have ACC. It's pretty common. Okay. So people have asked me about the individual who is fictionalized in this movie. So we'll just leave it at that. It's a movie. We all know about the movie, but behind the movie is a real person, or actually two really important people. One of them is this gentleman here, Kim Peek, and he did have autism in the sense that he met clinical criteria for autism in terms of his behavioral challenges. And that's his dad that he's talking to. His name is Fran Peek. Kim passed away a few years back, unfortunately. And there was a documentary made about Kim called The Real Rain Man. So I show this just to sort of make the point that there are individuals who have behavioral challenges that meet the criteria for autism, and it's a bit of a sort of check the box kind of approach to diagnosis. And at the same time, Kim also was born without his corpus callosum. And so that is true for a number of individuals with ACC, and we'll talk about that in a moment. But if you flip it on its head and ask how many individuals with autism have ACC, the answer to that is smaller in number. And I'll see if I can explain a little bit more about that as we go through the talk. Okay, so autism is seen in many individuals with ACC. Many individuals with autism have developmental changes that are subtler in their corpus callosum, and I'm going to talk about one genetic example on chromosome 16. And then I'm going to talk about the advances in autism genetics and how that, in a way, allows us to understand the interplay between ACC and autism. Okay, so we have been enrolling participants, and if anyone is in any of our studies, I thank you for your participation, and couldn't do any of this without their help. But we sent out a questionnaire, and this questionnaire was designed by an autism researcher in the UK. His name is Simon Baron-Cohen, and it's called the Autism Quotient. And the idea is that it doesn't say whether you have autism or not, but it says that you have challenges that are on the spectrum and that they should therefore be evaluated. And he's designed this to work for higher functioning individuals. So all of these individuals that were in our study, and you can see the numbers here, 35, 19, and 48, were high enough functioning where they could complete this survey, or in the case of children, the parents could complete it about them, to see if we could understand what their risks were for autism. Okay, so let's just go here. So this is the first set of results. So this is children with ACC, control children, and then children with autism. The last two groups were groups from Dr. Baron-Cohen's cohort, and the top group is the ones from us. And so let's just focus on the far right over here. And you can see that, get the arrow to work. Maybe I'll give up on the arrow and point. Does anyone have a pointer on them? So you can see here that of the children with a diagnosis of autism, 95% of them cross the threshold. So that's sort of like proving that the test works in a way. And if you look up here, typically developing children, 4.3% of them cross over the threshold. And so what that tells you is essentially the false positive and false negative rate of the test. It's not perfect, but it's in the ballpark, right? It's sort of what you would expect. If it were perfect, this would be 100%, and that would be 0, okay? So by comparison, children with ACC, 45% of them cross that threshold. So no, that doesn't mean that 45% of them have autism. It just means that using this particular measure, that 45% of them are, have symptoms that require further evaluation. So there's a question over there.
Elliott, I just wonder whether there's any correlation across the gender between genders in that, because Baron-Cohen's work has been rightly criticised for a gender bias towards his extreme male brain kind of thing. I wonder whether there's any correlation in your numbers in that regard.
Right, yeah, that's an excellent question. We had sort of an equal balance of boys and girls in our study, and I didn't see a particular boy bias, but that's an excellent question, and I agree with you that there is that intrinsic bias in sort of his approach. Okay, and then this is now looking at adults, And this is looking at, again, adults with autism, neurotypical control adults, and then adults with ACC, and then this is self-reported, right? So this is, the individual, him or herself with ACC, commenting on what they felt, were the traits. And again, this is not a population study, so these numbers can't be 100% extrapolated and say, well, therefore, by definition, all the individuals who are children with ACC, 45% of them cross the threshold. These are the people who happen to be in our study, who happen to fill out the form, right? So there are caveats to all of this data. Okay, but using that, you can see that it was 20% in the top corner here of individuals, adults with ACC, 79% in the adult control group, and then in the adult neurotypical controls, it was 2%. So sort of similar. There's, again, a lower percentage here than you would expect if the test was perfect. And so for both the adult ACC individuals and the adult autism individuals, it's probably under ascertaining. And then we did the same thing with the adolescents. And again, it was 88% for the adolescent ASD individuals, 26% for the ACC individuals, and 0 for the neurotypical controls. And this is another way of looking at the data. And so you get a score, and the lower the score, the more likely you are to have autism traits based on the way the study is designed. And if it's higher, you're more neurotypical. And so what is in red is the distribution of the individuals who are children who had ASD, and you can see their scores are shifted over to the left. The neurotypical controls are in green, and you can see that they're shifted to the right. And so the overlap between them is pretty small, whereas the ones that are in blue are ACC. And you can see that they're essentially similar, that there's a normal distribution, meaning that there's a middle and 2 tails, but that is just shifted over. And so one way of thinking about this is that ACC, like a lot of other things, is a risk factor, but not the only thing that accounts for whether somebody has a diagnosis. Okay. We did some other work where we tried to see if we could understand what connections in the brain might be explaining that. And this is a little bit of a complicated slide, but it's basically telling us that the area that is in blue, which is in the frontal lobe in the lower part on the right-hand side, that the activity of that area, when it was underactive, those individuals were more likely to have autistic features. And other people have found this same area, what's called the ventrolateral prefrontal cortex, as being an important area that signals for social cognition that we think is deficient in both individuals with ACC as well as individuals with what I would refer to as idiopathic autism. Okay, and then this is work taken from Dr. Lynn Paul, who's also here, and this is very nice work that she did. The benefit of this work over ours is that these were in-person assessments, and they were using a tool that's called the ADOS, Autism Diagnostic Observation Scale. So ours is a survey where people fill out the questionnaire. Hers is psychologists in a room with the person, interrogating them and coming up with a diagnosis. And again, you can see here that the neurotypical controls in hers are the red circles, the ASD individuals are green, and the individuals with ACC are in blue. And so if you focus here on the lower left, box C, you can see that and you have to cross the threshold where the 10 is to have a diagnosis, you can see that many of the individuals with autism, but not all of them, cross that threshold. And same thing with ACC. You have many individuals that are sort of at risk, which is the lower dotted line, as opposed to the thicker upper dotted line. And so the distribution in ACC, again, is not as robust as autism. Not everyone has it, but there is a significant percentage that do meet the diagnostic criteria. So this is complementary. This is only adults, whereas ours were adults and kids. But I think we're telling pretty much the same story. Okay. So one of the things that has happened in the last 10 to 15 years is that genetic tools have gotten a lot better. When I first started out in the field of neurodevelopmental disorders, there were just a few conditions, Fragile X, neurofibromatosis, tuberous sclerosis, that we knew were linked with cognitive and behavioral challenges. And in the last, again, the last 10, 15 years, there's been a huge transformation in our understanding of these different conditions. One of them is this particular genetic change on chromosome 16 that I'll refer to as 16p11.2. So what is 16p11.2? Well, geneticists love, let's see if I can get my arrow up here. Geneticists love numbering like the Dewey Decimal System a little bit, or it's more maybe like a street address. So this is chromosome 16. So this whole stretch here, and actually goes beyond here and beyond here. And each chromosome has many millions of letters of DNA on them. And then each chromosome is divided up into, if chromosome 16 is Melbourne, actually I shouldn't do that because I don't know the neighborhoods in Melbourne, but let's say we did San Francisco, where I'm from. If chromosome 16 was San Francisco, one of these little things here would be, the Marina, which is a neighborhood, and another one would be Pacific Heights, which is another neighborhood. So each one of these is like a little neighborhood, and in these neighborhoods, these genetic neighborhoods, sit lots of different genes. And so what we're focusing on is this region here that spans from what we call breakpoint number 4 to breakpoint number 5. And in that region, there are about 30 genes. And these are the genes that are indecipherable, I apologize. But the point is that this region, it's about 500 to 600 million letters of DNA. There are 30 genes in here. And the reason why these are called breakpoints is because it turns out that this stretch of DNA looks very much like this stretch of DNA. And you may remember from your biology genetics that if, when mom's embryos are made, when the eggs are made, the DNA undergoes recombination. And when the sperm is made, the same things happens too. And that's how, somebody might end up with, Grandpa Joe's nose and Grandma Jane on the other side's eye color, because, there's this swapping of DNA. But this swapping can also take place on the same chromosome. So not on mom's copy or dad's copy, but just on mom's or just on dad's. And that's what this is showing here. And that could lead to either a duplication of that chunk of DNA or a deletion. And that's what this little circle diagram shows. So we enrolled about, I don't know how much that is, almost 300 individuals to participate in a brain imaging study. This was done in the United States at 5 different institutions. And they came to one of the 5 sites, and they got brain imaging if they were able to hold still, and we did cognitive testing, and we did testing for whether they were on the autism spectrum. And what I just want to highlight is, really the numbers are very large. So there's 75 individuals who, for this genetic address, they only have one copy instead of two, mom and dad, so they just have one. And then there's another group of individuals, 70 of them, who carry the duplication. So they carry, instead of two copies, they carry three. And these individuals were identified to be at risk for autism based on having these genetic changes. Okay, and we also had controls. We had family members who were siblings of the affected individuals who didn't carry these genetic changes, and then
I didn't hear what it was. Sorry.
it's okay. Siri's very curious, and over 100 population controls. So again, individuals that were roughly the same age and gender as the group, but had a slightly different IQ. And so you can see that here, they're pretty well matched for male-female ratio, to come back to that earlier question. They're matched for age reasonably well, but not for all of them. And the IQs you can see are a fair bit different. The deletion carriers have IQs around 80, the controls around 100. Okay, but what we wanted to ask was, one of the many things that we asked is, do you see differences in the corpus callosum? Right, so now this sort of gets back to this question about autism. Is autism associated with disorders of the corpus callosum? If you read the literature, the answer is maybe. You read some papers where they say, yeah, we see it a little bit here, and you read other papers and they say, no, we don't see anything at all. And the reason why that is, that I would posit, is that some of the genetics of autism overlaps with the genetics of ACC. And where that overlaps, you're going to see similarities. But if you just take a group of, even a big group, 100 individuals with autism, in that group of 100 individuals, it's likely that there are 50 or even 100 different causes of autism all mixed in. But if you start with a group of individuals who have the same genetic change, in this case either the deletion or the duplication, that you see much more consistent findings. And so what this is pointing out, so again, here's a normal corpus callosum, and looking at it in the middle, and then here are two examples of individuals who carry the duplication. One person has a thin but fully formed corpus callosum. You've seen me show you that earlier. And then this one here is an individual with a very small corpus callosum. Those are both duplication carriers. And if you are a deletion carrier, you actually have the opposite. You actually have a thicker corpus callosum. And we wouldn't have been able to see this and see the significance of this unless we had all the individuals who shared that same genetics participate together. Okay, and so this is how the statistics line up. So if we're looking at a thin or smaller corpus callosum, approximately 20% of the duplication carriers had a thin or smaller corpus callosum, whereas population controls, only 1% had a smaller corpus callosum. So that's what it is in the that's the frequency in the general population. So you can see that that's a dramatic difference, and we wouldn't see that unless we line them up. And then if you look at the deletion individuals, how many had a thick corpus callosum? 16%, not quite as high, but still significant. And again, the population controls is 0. So this is something that is probably associated with the genetics of chromosome 16p. Okay. Oops. How are we doing on time?
Fine.
Okay. Okay. Now I'm going to switch gears a little bit. I told you about a chromosomal address that had about 30 genes on it. And that was done by a tool called a microarray. Can I just have a show of hands if your family member had a microarray done? Okay. About a third to a half. And then how about a show of hands if anyone has had exome sequencing done? So fewer, but probably a growing number. So exome sequencing, for those of you who haven't heard that term, is a way to sequence all the genes in somebody's genome. Not all of them, but most of them. And this tool, it turns out that the genes themselves, the pieces of DNA that code for the proteins, only make up about 1% to 2% of the total genome. And we, as doctors and scientists, know best how to interpret genetic changes that occur in the genes themselves. There could be genetic changes elsewhere in the genome, but we're not as sophisticated yet about how to interpret that information. So because sequencing was expensive, people developed a tool that allowed them to grab or capture the part of the genome that is in the genes and sequence just that part of it. And so what this represents, these are two figures from a paper by a colleague of mine named Evan Eichler, and his group sequenced a number of genes in many thousands of individuals with autism. And what did he find? So the first thing that he found is that there's lots of different genes. There are probably hundreds of different genes that cause autism, and there are probably just as many that cause ACC. The next thing is that, again, he's just looking at genes that cause either intellectual disability or autism. He's not interested in whether there are brain anatomy changes. I mean, he is, but not in the way he's done this study. And I'm just pointing out quickly here 3 genes, ankyrin2, which is highlighted in that oval, ARID1B, and DDX3X. It's probably hard to see that last one, but that's in there. All 3 of those genes are 3 of the most common autism genes when you just say, how many times do you see children with mutations in that particular gene of interest? All 3 of those genes, ankyrin2, ARID1B, and DDX3X, all 3 of them are associated with ACC. And I apologize, I'm going to jump through this a little bit quickly, but just to make this point. So again, I showed you 16p, and I showed you brain imaging changes that we could only see because we were fortunate enough to be able to recruit enough individuals in each of the groups to see the similarities that they share. So DDX3X is a mouthful of a gene name, and I apologize, I didn't name any of these. My wife still gives me a hard time for not having named one after her. And I'm not going to dwell on the biology too much because it's quite complicated, but what I would just say is that that last X is because the gene is on the X chromosome. And there are almost no boys who have a mutation in this gene. And the thinking is that since boys only have one X chromosome, if you injure it with a mutation to an essential gene, that the boys are never born. So, it would be some sort of, a mom, a woman might be slightly late on her period, and it would be something like that happening. But there are girls who are born with mutations in this gene. This gene went from essentially being unrecognized as a cause of developmental disorders to now having over 100 individuals identified with mutation in this gene. And probably literally every week I get 3 or 4 emails from families around the country and around the world where their daughters are diagnosed with this. And that's only because this new tool, exome sequencing, is available. So there are a couple of things that I want to highlight here. One is all of these little lines refer to different locations where mutations have been reported. And what you can see, just as an example, is that there's 3 letters, a number, and then 3 letters. And so the first 3 letters, in this case, stand for the amino acid arginine. and then the three numbers is at amino acid position 488, and then it says cysteine, CYS. So it's saying that the normal amino acid at position 488 is arginine. There's been a genetic change, and the predicted change would be this new amino acid, cysteine. So anywhere where there's blue or anywhere where there's red means that there are multiple mutations occurring at the same location. So what that tells me is that there are hotspots, that certain areas, when genetically changed, are more likely to result in developmental challenges than to be silent. Because we all have genetic changes in us, and they're mostly silent, just kind of hanging out. And then the other thing you'll notice is that there's a high preponderance of mutations in either this red domain that's called the helicase C-terminal, or in here, the helicase ATP binding domain. There are a few mutations in here, but not nearly as many, and there's essentially none in the tail of the protein. So again, there are certain areas that the protein probably can't tolerate these kinds of changes. Okay. So again, like the 16p case, because we have lots of individuals who've agreed to participate in the research, we can see different things. And I apologize, I'm going to not dwell on this because of the time. A number of them, but not all of them, have absence of the corpus callosum. But 29 out of 53 of the individuals who are in our study have a thin corpus callosum. So again, you wouldn't have been able to see this if you just had one patient show up in your clinic. But when you have 50 individuals, you can see a pattern, and you can see it very quickly. And then this is another change. This is what's called the lateral ventricle. Here's part of it, and here's part of it, and then the lateral ventricle is up here as well. So it's kind of like a C shape. It starts out at the top, and it goes down like that. And what you're seeing down at the bottom, where my thumb is equivalently, is that this space, you'll have to take my word for it, but this space is way larger than normal. And that's something that we see again in 30 out of the 53 individuals. So again, now I can see very characteristic changes of a disorder that's associated with absence or partial absence of the corpus callosum that I wouldn't see with just 3 or 4 kids. And then there's another kind of brain change that's called polymicrogyria, meaning many small gyri. And you can kind of, let's see if I can point it out. So if you look right here, it looks like the darker gray that is kind of on the surface is about the same thickness, and they're kind of large areas that are what we call the gyri, the curves on the outside, and the sulci, the deep little caverns. But if you look in this individual here in the front, or even better in this individual in the front, you see lots of smaller ones, and they look sort of jaggedy or almost like you've crumpled up a ribbon. And that's, again, a different kind of brain change. It's not as common as the other ones, but again, we wouldn't see it without having everyone to evaluate. And this is just sort of a schematic of what that looks like. Okay. One last thing. This is a biochemical assay. So there's a graduate student in my lab, and what he did is he made the protein, the DDX3X protein. He made a normal one, and then he made one where he was able to introduce a mutation. And one of the protein's function is to take RNA, which is double-stranded, so 2 strands are stuck together, and you have to be able to open it up for the RNA to function. And so that's what this helicase activity is. It's like an unzippering of the RNA. And when it's unzippered in this assay, it ends up being down here. And you can see this looks like just like a thin single strand of RNA. And then up here, you have the 2 strands. So if the idea is if you have a mutation, the expectation would be that you would only see double-stranded, not single-stranded. And if you had a protein that was working, you would see single-stranded. Okay. And so that's what these diagrams are showing us. The dotted line that's black is control. And so that's going from 0, it says fraction unwound, from 0 to almost 100% in under a minute or two. So that's what the normal protein's supposed to do. It works really fast. It's in part regulating cell activity. And then if you take this one here, this change from alanine, so A233V, so valine, you make that single amino acid change. Instead of it working at 100%, it's working, but it's a fair bit slower. So instead of working in under 1 minute, it takes about, I'll find that arrow right here, it takes about what looks like about 60 minutes to complete its activity. And then there's this other one here that's a T323I, so threonine 323 isoleucine. And those changes result in essentially no activity. So it's like the protein is completely broken. It doesn't work at all. And just to go back, these changes, this extra change, not the ACC part, but the extra change in this polymicrogyria correlates 100% with no activity of the protein. So when there's no activity of the protein, we see extra changes in this, in the essentially injury to the brain as it develops. Okay. So I don't want to leave you on a totally depressing note here. One thing I will say before explaining what this slide is that, science is geared toward finding differences. So that means we can measure how the normal brain works, and we can measure how changes in that normal brain work. And so it's much easier for us to see more dramatic changes than it is for us to observe and understand subtle ones. Just so you don't leave here concerned. Now, this is something, there's AusDoC, there's the NODCC, and those are fantastic organizations, and I applaud their purpose and their goals, and I will do whatever I can to support them. One thing that's happening in parallel, and I think will hopefully be synergistic, is support organizations for single genes. So, for single genes, there are enough families to make an organization, and so this group here is families where their children have mutations in DEX3X. And at the time that I took the picture of the web page, they had 95 known cases and almost 70 families signed up. And they've only been at this for about 6 months now. So they're making really strong progress. I just want to point out, where's Tim? Here's Tim Edwards. He's getting his PhD in Linda Richards' lab, and he spent some time with us, and I'm one of his co-mentors. This is Brianna Ferjot. She's one of the clinical coordinators in my lab, and if you reach out to anyone, you would probably hear from her. This is Jung Lee, the senior scientist who did a lot of the biochemistry that I showed you. And thanks for your attention.
So I began by looking at your program and your theme for your conference of RAW, and it really made me think that this is probably pretty much where the families or the parents of autistic children back in Melbourne in 1967 were thinking when they started Autism Victoria, which it has had a number of name changes. So I wanted to say to you, congratulations coming together, because this is where all great movements and things start. So what happened, and I've been reflecting on, and I say this often to people, that autism is probably relatively pretty new, in the scheme of health and disability. And so I was looking back and found that it was in the 1930s that a scientist or a clinician called Leo Kanner actually defined autism as a condition. And everybody knows that in that period of 37 to 45 there was a big world war, and research that was going on in Germany by a man called Hans Asperger was pretty much kept quiet because nobody was doing too much with the Germans after the Second World War. So Leo Kanner did become sort of the guy in autism and named it. But we don't think a lot about Leo Kanner because he said the cause of autism was largely due to refrigerator mothers. That meant that mothers that were very cold and not loving towards their children was what was causing autism. And Leo Kanner was involved in a lot of what we call aversive therapies of treating autism back in those times. So AMAZE is not a huge fan of Leo Kanner necessarily. Who we are a huge fan of is Margot Pryor, who is our patron, and Margot is a wonderful researcher and clinician, and she wrote the first paper on autism in Australia in the early 70s. So if you think about AMAZE, a group of parents coming together to RAW, to have resources, recognition, access, etc., Margot wrote that paper a few years after that. So it's really, the last few decades that we're talking about. Autism itself in the DSM-3 was first differentiated from childhood schizophrenia, and in 1981 Asperger's syndrome was recognised, and again it was the first time that Hans Asperger's work was actually translated from German to English, and we actually found out about it. So that's just a quick snapshot. Where we are today, autism in Australia, we say 1 in 100 people in Australia on the autism spectrum. Boys to girls ratio is now 3 to 1. Only 2 weeks ago I would have said 4 to 1, but some new research came across my desk this week that's showing it's actually, some meta-analysis was showing it's more like 3 to 1. And we are hearing lots from girls like Penny and an organisation, one of 3 organisations she's representing today, Yellow Ladybugs, is just, as women do, driving this whole thing about women and girls with autism. It's the largest primary diagnostic group within the NDIS, and by that I mean 28% of all plans, funded plans for NDIS, have been written for people for whom autism is their primary diagnosis, and that is way above any other condition, so it's there. Some research I'll talk a little bit more about that we conducted was, we found just last year some research that 52% of Australians have had or currently have a personal connection with autism. That means they have somebody in their family, they have a friend, they work with, they went to school with, whatever. So that's pretty high, half of the community. And 35% have or know somebody with a family member. The downside or the bad news that we're working to improve is 35% of autistic students achieve only year 10 or less, compared with, so it's double the rate of that in the population, and only 46% of autistic adults are employed versus 43% of the population. Yeah.
The ABS data is actually just coming out saying it's actually 41%.
41, thank you. Is it because
41%, and that's of any employment. It's not full-time employment.
yeah, any employment. I'm sorry, and I thought I was actually quoting the 2015 study, but I might not be right.
The diagnostic workshop was held by Autism TLC last week.
Right.
They put up the 41%.
41, yeah. I'll have to challenge Andrew Whitehouse because I think it's 46. But anyway, I'll check with him. Thank you. We had lunch after that workshop, so he didn't mention it. So I'll see. So I came into AMAZE about 3 years ago, and really what I want to talk to you about is what we're doing for what we believe our role is and our purpose in what we're going to do for our community. So we call ourselves the peak body for autistic people and their families in Victoria. And so when we were developing our new strategy and purpose, so there were 2 main drivers, and really the first one was the laws of the land, both internationally and in Australia, that the United Nations Convention on the Rights of People with Disability was, Australia signed up to that in 2006. We have our own Disability Discrimination Act, and we have disability standards for education. So all these laws are really important because they make a mark that says to the world, we have to treat people with disability as equitably as with anyone else in the population, and that, as we know, has not been happening. So that's very important to us at AMAZE, but also more importantly was the feedback from our community. We had 600 people tell us what they expected from us was firstly to listen to autistic people, raise awareness of autism, work with governments to influence policy and provide support and information to our community. So that's pretty much what we've agreed to do going forward. So we have written a 25-year strategic plan. Our belief is that it takes generational change for us to achieve the social impact. The impact we want to make on the world is that we have a society that respects the dignity of every autistic person and provides them opportunities to participate and contribute. That is really what our community was telling us, that they have the right to do that. And again, what we do, we build awareness and understanding, and we want to develop the capacity of the wider community to support autistic people and their families. So our 3 goals, as I said, increase understanding, influence change and skill up the community. And really the organisation, as I said 50 years ago, really started with working with autistic parents of autistic children, then more to autistic people, but today we say we're working with both, but really the focus is on the wider world, because our belief is it's not just for autistic people and their families to change and to fit into the wider world, it's about the wider world changing and skilling up and stepping up to support autistic people. So how do we do that? So I mentioned that our community said we need to raise awareness of autism, but we've come to understand that you could ask the average person on the street, have they heard of autism? And from Rain Man and other things, people will say they've heard of autism. We don't have a problem with saying, have you heard of autism? But what we really don't know, or what we really need to do is make sure they understand what autism is, dispel some of those myths, so whether we get, I'm autistic, oh, you must be like Rain Man, you must be a genius, I'm autistic, and certain misconceptions about being autistic, and also, but most importantly, how do we skill up, again, the wider society to support autistic people? So some of the research, again, we found, so just around facts and myths, we were actually pretty pleased to find that 74% of the community agree with us that there needs to be more understanding in the community about autism. They understood that autistic people were under or oversensitive to light. They know that autism, the incidence of autism is growing, although that was about only 4 in 10 people. They do know that no 2 autistic people experience autism in the same way, so around 2/3 of people. They know that anxiety is very common in people with autism, which is really great. And then on these myths about autistic people can't make friends and lack empathy, again, only 1/3 of people thought that. So that's pretty positive. But again, they didn't know the number of girls with autism is increasing. So it gave us a feel for what does the community know. If we're going to increase understanding, we have to find out what their level of understanding is first. And in terms of supporting autistic people, 72% of people believe that schools, workplaces and the wider world needs to have a better understanding of how to support, but personally only 1/3 of them said, I personally would know what to do to support an autistic person. So this is really where our work must focus. We're going to be doing more research in this area. So if we want to change community attitudes and build support for autistic people, we have to know what the attitudes are and be able to track them over time. So we're currently in the field conducting 2 studies, one around community attitudes, so that's 2,500 Australians have participated in that study, and then we're doing a companion piece of research that is what are the experience of autistic people in their families. So the community might be saying one thing, but autistic people themselves. And then we will publish those and promote the findings. How do we promote the findings? This is 2 television news interviews that I've done, so very much about if we can build understanding and build a better world for autistic people if we're out in the media talking about it. So talking about what our priorities are in terms of education, employment. We want to change those education results. We want to change those employment results. And we need to see more positive portrayals of autism. So we're always hearing about violent autistic people or children in schools being put in cages, or all this negative portrayal of autism, and so we are very focused on creating some positive portrayals of autism. One of the, as I said earlier, one of the key things our community said was you need to listen to autistic people. They need to be engaged in the work that you do, and they need to be able to tell their story. So I see AMAZE very much as an organisation that is going to facilitate the voices of autistic people being heard. I don't need to speak for autistic people. They can speak for themselves, and Penny here has spoken for us and herself on many occasions, and will today. But this is a campaign that we've run for the 3rd year now on World Autism Awareness Day on April 2nd, called Spectrospective, which is Stories of Autism. And if we have time at the end, I'm going to show you just a clip from this year's. But that has now, so we've made 3 films of autistic people's stories where they've told their story in their own way, and more than 100,000 views of those videos that have been made. So again, autistic people can say it best about what they need and what they want the world to be. So secondly, in influencing for systemic change, so by this I mean the A word is advocacy, which tends to be not a very appealing word to conservative governments, so we sometimes say we're influencing, but we really see ourselves as advocates on behalf of our community to change schools, to change workplaces, to change the wider world. And so we will work very strongly with government and advocating to government that we have a policy area at AMAZE that we're writing a number of submissions to the government. So in this year, you may know, there was a Victorian parliamentary inquiry into autism, and we had submissions from 450 autistic members of our community, autistic and parents in our community, that made submissions into us that we put into this document called Driving Change. We also had were very active and at the table with the state government's review of the program for students with disability, which is all the funding and aids that students get in schools. So we've been very active in what needs to change in schools. And there's currently the Productivity Commission, you've probably heard, is reviewing the NDIS. Again, we had about 150 submissions from our community to feed into that. So again, we want to present the voices of our community to affect change. So in terms of building the skills of the wider community, we provide a lot of information and support to autistic people and families, but also to the wider world. We have an Autism Information Advisor telephone support, and we have about 8,000 calls to that every year. So you can see we are providing a lot of information support, just as the founders of our organisation 50 years ago really wanted us to do. Our website is very well accessed, and what we pride ourselves on is our information is credible, independent, evidence-based, but also we are treatment agnostic. We don't say to any family, this is what you should have or not have, like a service provider might. Our role is to tell people what is available and for them to make informed decisions. We also hold a number of events and knowledge building programs and a lot of forums. We are currently running some NDIS workshops for support groups. We have early days workshops, which is when you have a parent's children are diagnosed, they can come along and meet other families. And also we held our conference last year, which Penny spoke at, along with 25 other autistic people. Out of 50 speakers, we had 25 autistic speakers, which was amazing. And we had about 500 people attend that. So AMAZE has always run the Victorian Autism Conference every second year. And last year's conference, I said, was different in that the conferences before had been about autism, but this conference was really for autism and for autistic people to have their voices heard. So in terms of the wider world, we also do a lot of consulting to organisations. For example, Northland Shopping Centre, you might know, is the first shopping centre to our knowledge in the entire world that has a quiet room for autistic people. And that room and the creation of that room was driven entirely by a mum that worked in the centre management there. So it's amazing about what parents can do. So that quiet room is for autistic people who work at Northland or who visit Northland, and they also have sensory Santa Claus, and they have shopping times where just autistic people can go. So, I mean, I'm not autistic, but I know a shopping centre environment is pretty intense for me, but it doesn't stop me going there, whereas for an autistic person, they may not be able to go there. So that's been fantastic. And hundreds of people are using that quiet room, which is fantastic. Museum Victoria, so that's the museum up here in Carlton, Science Work and the Immigration Museum now have autism-friendly policy in their organisation. They have a sensory map that will show high and low sensory areas in the museum, and that map had been downloaded something like 8,000 times. It's just astonishing. So it's really great, and also with Parks Victoria, we've been working. Village Cinemas were the first to introduce sensory-friendly screenings, and they're a partner with us around Spectrospective and show the film every World Autism Awareness Day. Again, thousands of people have attended that. So it's really good that we are seeing the wider community recognise, and it's good business for them to recognise that they need to be providing support for the diversity of their customers. So Spectrospective I mentioned, and I would like to just play a short clip from this year's Spectrospective, but also to say to you that you can stay in touch with AMAZE and what we do. This is our free e-newsletter called eSpectrum that goes out every fortnight. We have about 15,000 people signed up to that, and we'd love to have you also join that. So, I will leave you, as I should, with the voices of our community. Thank you. Oh, here we go.
A different way that our brains work.
I would get really anxious.
When there's too much going on in the brain, it's like you sort of fill up the tank and then it sort of overflows.
I spent 40 years not knowing what on earth was going on. I am autistic.
Welcome to the Spectrospective.
What does being autistic mean to you?
It means having its challenges and different ways in life.
As a carer and as a spouse for someone who is autistic means just a variety of colours, a rainbow.
Autism is a spectrum. Our three children are all completely different to each other.
I love doing art.
I love sewing, and this is the cow I made last week, and it took me a day.
This is my new double-sided sequin fabric.
My biggest passion overall is music.
I like the fact that I'm incredibly honest, and I mean, I'm not tactless or anything, but I will say what I need to say, and I work on one level. I only operate on one level. I find one of the difficult things with communicating with non-autistic people is that they don't a lot of the time, and so there's a lot of hidden meaning going on.
Sometimes being that person that just doesn't understand the joke, I don't really get his joke sometimes. I think they're just strange.
It's been a massive journey for me of living with a lot of trauma and shame, thinking there was something terribly wrong with me. Why couldn't I succeed in ways that I really wanted to? Why were people so difficult? Why was work so difficult? Why was the world so difficult?
Being diagnosed as an adult, it was quite relieving to me because all of a sudden the rest of my life just made sense.
I actually really love that there's so many organisations to help with it.
Bringing Chrissy and the service dog into life has made it a lot easier to go out in public, go to shops, go for walks, things like that.
I started Masters this year, and I'm working really hard on how I can make being autistic more socially acceptable.
Autistic people can work, but they are not given a chance because autism is still seen as a collection of symptoms, collection of disabilities, collection of difficulties, as opposed to a person, a full person with some difficulties.
If words aren't their thing, then watch. How do they process the world? How does information come into them?
I'm a great person, I like me, you know, I've actually, after many years not liking me, I like me and I'm happy to be myself, so I'll wear the coloured wigs, you know, I'll get up on stage in the flowery shoes.
My life is a lot more richer and a lot more colourful because of the amazing autistic people that I have in it.
It's really important to do the things that make you scared because you love them, and to overcome that fear and tell fear, you know what, you're not going to hold me back from doing anything that I love.
another round of applause for Fiona.
So, as people have already said, I'm the wearer of a few different hats, and I've only had my AMAZE Victoria cap for about a week. So now I can finally do stuff and not be bothered by those annoying lights. The ICAN Network put the awesome into autism, and I have to have the spelling on the board, otherwise people just don't get how we spell it. And yes, this is definitely a presentation where phones out for taking photos is probably necessary part way through the talk. So Elliott and Fiona have been talking a bit about autism, so I thought I should firstly say what autism means for me. It gives me positive things like attention to detail, honesty, like Jeanette Perkins said in the Spectrum video, and being conscientious. But it also means that I can be anxious. I have sensitivity to light and sound. So here's a photo that I took on the plane in full autism uniform with my sunglasses on because I had a window seat. I love looking out the window, and I had my noise cancelling headphones on. So I certainly cope better on flights now that electronic devices are allowed. Music all the way. But as Fiona has said, each person with autism is different, and over the month of April, the ICAN Network featured a different person's story every day. So if you go to ican.network/humans, you'll be able to read other people's stories of being on the spectrum, including Abby, one of the other people in AusDoCC. So here's my brief story, and then I'll talk about yellow ladybugs. When I was a young child, my speech was atrocious, and my parents tell the story of once I wanted pizza for dinner, and it wasn't until I was doing the hand waving of throw the dough up and then catch it and roll it that my parents finally figured out what I wanted. So I had speech therapy in 3 and 4 year old kinder, and then in school as well. And I was bullied a bit at school, especially in years 7 to 9, when the kids' "you're different" radar was at their highest, because of course I was the nerd, the square, who did all of her homework. And I was teased a bit for my lisp in year 6, and I was self-conscious about that. And then when I was in year 12, I started getting panic attacks in the school assembly hall. And that would mean that I'd be sitting in the audience of the assembly hall under no danger whatsoever, but my palms would be really sweaty and my heart would be beating really fast, even though I was under no danger whatsoever. And the first time it happened, I walked out of the lecture theatre, like the assembly hall, felt fine. So then I walked back in again, and guess what? The symptoms returned. So luckily my school counsellor knew what was happening and was able to tell me that's panic attacks, and just knowing the label allowed me to put some coping strategies in place. And one of these was sitting on the end of the row when I was in uni lecture theatres. But now I'm actually a lecturer at Monash University, and I lecture up to 400 students at once. And this is my view of the lecture theatre now. And this is taken in C1, which fits 300 students. This year will be in South 1, which fits just over 400 students. The joys of having 750 in your class. Have to do everything twice as well. But when I was diagnosed with Asperger's at the age of 14, I didn't actually meet anyone else like me until the end of first semester uni, like nearly 5 years later. So I went to my first support group meeting, Asperger's Victoria, just before I started working about 7 years ago. So then I became involved with the ICAN Network and Yellow Ladybugs because I had no support as a kid as I was growing up. And I knew I was that different kid, so I would have liked to have known other people who were also that different kid. So Marie's asked me to talk briefly about Yellow Ladybugs. Yellow Ladybugs was formed by Katie Coulis about 2 years ago when her younger daughter Mia was diagnosed with autism. And like all mums do, when you come back from the paediatrician and you've got the autism label, you do a Google to find out more information. And she couldn't find any information about support groups for girls. And Mia, who's her younger daughter, A, was obsessed about Yellow Ladybugs, and B, didn't get invited to any birthday parties in prep. So she decided to start up a support group for girls on the spectrum, and at the moment it's girls on the autism spectrum attending mainstream school, because we feel that people who go to a special school are more likely to see other people like them at their special school, and we can only hold so many events and they have to start somewhere. So as you can see, the mission is to improve the lives of autistic girls and women, and the vision is to be the leading Australian support network for autistic girls and women. Their tagline is "Every girl needs friends." At the Yellow Ladybugs panel lunch that was held just 2 days ago, one of the panellists turned the well-known song on its head, and her second slide was "Girls just want to have friends." And they also deal with some advocacy about the current situation, because girls are often missed, so they fly under the radar. They're tougher to spot because girls and boys present differently. So this means that girls are often misdiagnosed, or if they are diagnosed, they might be diagnosed at school instead of being diagnosed in the kinder years. So they miss that magic 7 early intervention number. And girls can also be misunderstood because they don't lash out. They tend to hold it in at school, and then their poor parents cop it after school. So as a result, they can miss out on funding, and they can also miss out in intervention at school. So Yellow Ladybugs hold all sorts of different events, and we also have the Go Yellow campaign on the 2nd of April, which is designed to shine a light on the girls on the spectrum as well. These photos, well, we've got Emma Yellow Wiggle on board, and she posts on Facebook each year, which is great. And we've got some photos of the picnic that we had last year at Melbourne Star. We've also held events at the gallery and the museum. Bounce and Inflatable World were my favourites because I could join in with the kids. And they also do some advocacy. I've already shared this image on the Facebook page of the conference because I know that it's a bit too hard to read all of that really small. And we also made it into the Herald Sun this year, and Katie also goes on TV and gets interviewed sometimes. I've made a really good friend in Lucy and her mum Natasha. So last night I sent her mum a quick message on Facebook saying, "Lucy, what's the best thing about Yellow Ladybugs?" Lucy, who's now 8, wrote back, I like my yellow ladybug bear, Bridget Bear, because she is a yellow ladybug just like me. They had a Build-A-Bear workshop earlier in the year. And Natasha said, yellow ladybugs gives Lucy a sense of belonging and a place to celebrate her autistic identity. And she's actually having trouble with school refusal at the moment. And when Lucy finally returned to school for like only an hour, when she came back home, yellow ladybugs had actually left a couple of yellow plants on her doorstep, which she loved because she loves gardening. And now very quickly I'll give you some resources. There's a great YouTube video called A Child's View of Sensory Processing that explains the 7 different senses. And yeah, quickly get your phones out now. And while people are taking phones out, I'll say I also have got various things over here. I love my tangle. Other people like mazes or these things, and there's also various weighted things. So some of my strategies include my Nui bag, my weighted blanket, my tangle. Oh, and don't tell my grandma, but I'm wearing my black and green sneakers today, not my black shoes. She won't be very impressed. One thing that I absolutely hate are the noisy Dyson hand dryers. I see a couple of nods. So one thing that VAC did was they turned off the hand dryers and had paper towels. And at APAC in 2015, I became known as Hoodie Girl after an incident with the really bright fluorescent light. So I put my hoodie up as I gave my talk. The cap looks better.
Can you think back to the hand dry one? Is that okay?
Yes, sure.
That's a very good visual representation.
Yes. We're running into afternoon tea. If you just want to take a couple of photos and delay afternoon tea, that is fine. And to all of the parents, I think it's important to quote Professor Rita Jordan from the first Victorian Autism Conference that I went to. The biggest gift you can give to people with autism is time. I believe that everyone has their own journey and their own timeline. So don't be afraid to do it your way in your own time and take lots of small stepping stones to achieve your goals rather than fewer, bigger, more daunting ones. So I did Year 12 over 2 years. I did my Masters over 4 instead of 3. So that meant that I was 27 when I started work. And then I lived at home for 3.5 years after starting work and finally moved out into my own place at the age of nearly 31. I'm 30, and then only 15 months later, my twin sisters, who were 7.5 years younger than me, had both moved out at home as well. So just the age of me and my other sisters moving out shows that each of us have our own timeline. And my grandma has been an extremely big support of me as well as my parents. So to quote Josh Gorban, "You raised me up to more than I can be." Because what we really want for all of our kids is to reach their full potential, whatever that might be.